Hi everyone, I'm Megan and I'm here to introduce our topic, which is tissue engineering in relation to the pancreas. Not many people know, but the pancreas isn't actually an organ, it's more like two glands. There's an endocrine part and an exocrine part to the pancreas, and it's about 6 inches long and it's positioned underneath the stomach. Originally it was Claude Bernard that discovered the function of the pancreas, and before he discovered the function, but diabetes was unrelated to it, like they couldn't link it up to diabetes at all. But he linked pancreas up to diabetes, and that's where the relation comes in today. Pancreatic cancer is one of the most common cancers, and it's responsible for 7% overall deaths in men and in women. And it's hard to distinguish pancreatic cancer as the symptoms aren't as distinctive as they are in other cancers, there's a high death rate. There's two parts to the pancreas, as I mentioned before. The exocrine gland, because it's made up of echinor cells, as you can see in the picture. This is just a zoomed up image. It produces enzymes into ducts, which mixes with bile and the duodenum, becomes pancreatic juice, which helps emulsify fats for digestion. Sometimes tumours can occur in these ducts, which cause blockages, and this leaks digestive enzymes into the pancreas instead of into the neighbouring ducts, which causes pancreatitis. The islets of Langerhans are small islands of endocrine tissue scattered around the cells. They make up the endocrine part of the pancreas as they secrete hormones directly into the bloodstream, like insulin and glucagon, which regulate blood sugar levels. Insulin is produced by the endocrine part of the pancreas because the hormones are directly released into the bloodstream, as I mentioned before. Diabetes is a condition where the pancreas doesn't produce enough insulin or doesn't produce it at all. It acts as a key by letting the glucose from foods like carbohydrates into the cells to produce energy. If insulin isn't produced, then the glucose has nowhere to go and it stays in the bloodstream, which results in dangerous levels of glucose in the blood. Insulin injections are given at regular intervals and in specific amounts to avoid a spike in sugar levels. Thank you for listening.
Hi, my name is Lauren and today I'm going to be talking about signals and scaffolds in application to the pancreas. So examples of signals are growth factors, peptides, DNA, miRNA, siRNA, mRNA, transcription factors and mechanical signals. So growth factors are necessary to ensure that the scaffolds have degraded and that the cells have grown and no longer need the scaffold. Scaffolds are used to support tissue formation, also needed to provide a template to support the growing cells. Over time, the scaffolds reabsorb into the body, leaving behind the natural tissue. The most suitable material for tissue engineering in the pancreas was seen to be chitosan. So chitosan can be seen here in the images. This is a zoomed up image of the chitosan. Desired properties in a scaffold is porosity. According to a study, the impact of size in pancreas islet transplantation and potential interventions to improve the outcome, islets of Langerhans need between 150 to 300 micrometers of pore size. So the pores are very important to ensure the diffusion of water and nutrients and vascularization. Desired properties are biodegradability. The scaffold needs to be biodegradable by surface erosion rather than bulk erosion. If a scaffold broke down by bulk erosion, the natural tissue wouldn't have a chance to grow. Another desired property would be the surface charge. So positively charged surfaces are more favourable for cell adhesion in the pancreas. Wettability. Hydrophobic surfaces absorb more proteins, and the texture. The rougher the pancreas scaffold is, the easier that the cells can attach.
My name is Lauren and today I'm going to be talking about the bioreactor that is used to grow the islets and a case study in relation to the pancreas. So first of all, on the screen behind me is an image of the bioreactor most commonly used for the production of islets of Langerhans. It is a vessel called the hollow fibre perfusion bioreactor. It is used for 3D cell culturing and it contains small hollow fibres, which is in the pink strip on the photo, that act as semi-permeable capillary-like membranes. The fibres are arranged in bundles inside tubular polycarbonate shells, which create hollow bioreactor cartridges in which the islets can grow. Cells are seeded into the extracapillary space of the fibres, which is the yellow portion of the image. Cell culture medium for growing the islets is pumped into one end of the cartridge, so it is pumped in through here. The medium provides nutrients and oxygen for the islets to grow and thrive outside of the fibre on the extracapillary space. In the extracapillary space, the yellow portion of the diagram, the secreted protein can accumulate to a concentration of up to 100 times higher than the conventional flask or roller bottle culture. So behind me are 3 images of a study carried out into the tissue engineered islets of Langerhans in rats. So the researchers used Chitosan sponge to mimic a capillary network. 20 rat islets were isolated and seeded into the Chitosan sponge. They were cultured for 62 days using DMEM and MEM cultures. The results were insulin was released constantly for 49 days from these islets cultured into the Chitosan sponge. However, after this, the research showed that they displayed a rapid decline in secretion levels for the rest of the study. So the top 3 images, A, B and C, are 3 photos showing the rat islets at various magnifications. The cells are stained with DTZ, which is dithiazone. They became crimson red because of this stain, and image C shows a lot of degradation, and the arrows are pointing towards it, and this is due to collagenase activity.
Hi, my name's Kira, and I'm going to be talking to you about the conclusion of our project and then the future direction. So in conclusion, Megan started our presentation by explaining what the pancreas is and its functions, explaining the difference between the endocrine and the exocrine functions, and how it produces insulin, which allows us to use glucose efficiently. And here you can see a nice diagram of the pancreas and its positioning in our bodies. So Lauren then discussed signals and the role they play in scaffold production. She went on to describe the desirable properties of a scaffold that would allow the growth of the pancreatic cells. Lauren O'Sullivan talked to us about the role of bioreactors and how they help us produce cells. She also explained a study conducted where islets from a rodent were isolated and grown in vivo. So, the future direction. Pancreas bioengineering lags behind other organs in tissue engineering fields. Only a handful of studies report successful repopulation of decellularized pancreatic extracellular matrix. The interaction of laminins and growth factors require further investigation. It remains unclear how growth factors augment yield in insulin response. The main issue impeding the development in this area is that when islets are implanted into a host, it causes an inflammatory response and there is no renewable cell source. Although in regards to the pancreas, bioengineering, a scientist recently made progress in the regeneration of a 3D pancreas. Exocrine and endocrine cell suspensions were perfused through a hepatic portal vein and successfully decellularized mouse pancreas. The resulting construct showed upregulated insulin gene expression that the authors attributed to the extracellular matrix interactions. Nevertheless, progress is impeded by the unanswered questions that remain in the field. Thank you very much.